Okay. You're welcome to today's class. Subject we look into today is biology. And it's been a long time. After how many weeks is this strike or break? What do you call it? Break. Suspension. Alright, so we'll start from we look at a very important aspect of biology. We are following the JAMB syllabus. We look at evolution among plants and animals. Evolution among plants and animals. So what we are going to look at under this topic is the subtopics we are going to look at under these topics are as follows. We look at the five kingdoms. Five kingdoms. Look at the kingdom Monera. The kingdom Monera, which is also known as the prokaryotes. An example of them, we have the bacteria and blue-green algae. Bacteria and blue-green algae. Then we also look at kingdom Protista. Kingdom Protista. Under this kingdom, we have those that have animal-like features and those that have plant-like features. Those that have animal-like features, we're talking about the what? The protozoans. And those that have plant-like features, we're talking about the what? The protophyta. So under those that have animal features, we have something like amoeba. Amoeba is an example. Then under protophyta we have paramecium. Even though we have the one that lies between the two of them, we have the euglena. Excuse me. Okay. We have the euglena. So these are things we look at under this topic. Then another kingdom we look at is the fungi. And under fungi, of course, we have mushrooms, rhizopus, and what have you. Mushrooms, rhizopus. And we also look at the fourth one. That is the kingdom Plantae. That's the plants. Kingdom Plantae. Under the kingdom Plantae, we look at these different groups. We look at the Thallophyta. Number one, the Thallophyta or the Thallophytes. Example, Spirogyra. Then we look at the second group we look at is the Bryophyta. These are under plants. Bryophyta. These are the mosses and the liverworts. Mosses and liverworts. Example, we have the Brachymium, that is Bryachymeni. Let me get the marker. This board is just to class, so you have those on the points and the sources. Yeah. Check if you can get something. Maybe this board is dirty fast. Maybe write something. If you don't see, don't worry, just come. Let's continue. All right. Listen. No, let's manage that. Maybe tomorrow. Please try and put the ball in order. Alright, so remember we're talking about the topic evolution among what? Plants and what? Animals. Among what? Plants and what? Animals. So, we said we are under what? Okay, what led to this is bryophyta, right? Mosses and what? Liverworts. So we have this. We have this. What's this? Pronounce it. Pronounce it.
Brachymenium.
Brag what? Brag what? Many.
Brachymenium.
Okay, we also have. What on this one? Mention what?
Sancho.
Yeah, good. So these are under what? Examples of what? Bryophytes. Before we get there, I want you to look them up. Know what they actually are, okay? Under what? Bryophyta. Bryophyta. Oh, these are mosses and what? Liverwort, right? Okay, so the next group I want to look at is the Pteridophyta. Pteridophytes. Pteridophyta. Pteridophyta. Pterido what? Fighter. So this is where we have our ferns. Ferns. They are what? Known as what? Ferns. Ferns. Example is what? Dryopteris. Example is what? Dryopteris. Example is what? Dryopteris. Okay. So now we'll look at the fourth one. Remember we started from where? Under plant, pteridophytes, right?
Yes.
Which are what? Example is what? Prosperidia, right?
Yes.
I mentioned it here. Then Bryophyta. This one is Pteridophyta. The third one. These are what we are going to look at under plants. Then the next one is what? Spermatophyta. Spermatophyta or what? Spermatophytes. Spermatophyta or simply what? Spermatophytes in English. So these Spermatophyta, under Spermatophyta, we have the what? The gymnosperm and what? Angiosperm. The gymnosperm and what? Angiosperm in English or Latin. Gymnospermia, okay? Spermia and angio what? Spermia. This is Latin. Spermia or gymnosperms and what? Angiosperm in English. So under gymnosperms you have the cycads, conifers are under what? Gymnosperm. The cycads are what? Conifers are under what? Gymnosperm. Then under the angiosperms you have your monocots, dicots, the monocot and what? Dicotyledonous plants. Example of the one in the monocot, you have your maize. You have your maize, dicot. Example, water leaf, and by the time we go deep into this, we now know the difference. Why these are called angiosperms. Which of them are flowering plants? Okay?
Yes.
Am I communicating?
Yes.
So I'm just listing what we're going to cover on that topic. Let's go into that. You know, the one that is that have one cotyledon and two cotyledons, the one that have let's not go there. We'll discuss that, we won't go there deeply. So these are what we're going to discuss under the kingdom Plantae. Now the last kingdom we'll look at is kingdom Animalia. Kingdom Animalia, or animals. Kingdom Animalia. Kingdom Animalia. Kingdom Animalia, or simply animals. Animalia, Latin, animals, English. So, under Kingdom Animalia, we look at the invertebrates. Look at the invertebrates. Look at the invertebrates and the vertebrates, okay? So, under invertebrates, you have your what? Coelenterics. Coelenterics, example Hydra. Coelenterics, example what? Hydra. Coelenterics. We have Kingdom Animalia. Kingdom Animalia. We have the invertebrates. Under the invertebrates, we have their what? Coelenterics, okay? Here we have their what? Hydra, example of them, the many of them. C and M ones and what have you, but let's just limit it here. Then under that, under one we look at Platyhelminthes. 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 A platyhelmint. Anyone? Platyhelmint, as case may be. Here we have your flatworms. Your flatworms, example, your tapeworm. That is the Tenia. The Tenia solid, Tenia saginata, as the case may be. Let us not go deep there. We'll keep treat this one after the other. So we have your Tenia. That is flatworms. Then the next one we look at, we are talking about the what? Invertebrates. The next one we'll take a look at is nematode. Nematoda or nematodes. Nematodes are the roundworms. This is the flatworm. Flatworms. Number 3 is what? Nematoda or nematodes, English. They are the what? Round worms. They are the round worms. Another one we look at is Annelida. Annelids. That's where you have your earthworm. Annelida or annelids. That's where you have your what? Your earthworm. Earthworm belongs to what? This Annelida. Okay? Where you have your earthworm. The next one under invertebrates we look at is the arthropoda. Arthropoda or arthropods. Arthropoda or arthropods. Of course, you know that under arthropoda we have different classes. We go deep into that. I just want to have a rundown of what we're going to cover under this topic. By the time we are done with this, at least questions come out from this in JAM. They don't joke with it. Two of us. So we're going to dismantle the one after the other. Okay, so we have here what? Arthropoda, we have here millipedes, name them, millipede, ticks, mosquitoes, blah 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 blah, cockroach, they are under what? Arthropoda, housefly, even though this is I listed that they are all under different classes, don't you know? They are not the same class. There are some that are what? Insects, okay? Some are distant. Cockroach, we are okay. Let me not take it far. Okay, now, so we have, after this, we have we also look at the mollusk or the mollusca, okay? The mollusca. We look at the mollusca or the mollusk. We look at the mollusca or the mollusk, as case may be. Now this is for the what? Invertebrate. So now let's look at vertebrate. What I'm going to cover under vertebrate. Under vertebrate, we now look at Pisces, that is the fishes, okay? We look at Pisces, or Pisces, as case may be. We look at both the cartilaginous fish and the bony fish. We also look at the second one, amphibia. Amphibia, we have your toads, your frogs, your toads, your frogs. Under what? Amphibia. Then we look at the reptilia or reptiles. The reptilia or reptiles. Look at reptilia. We have here, we have the lizards, snakes, even what? Turtles. They are not what? Reptiles, reptilia. Then aves. We look at aves. Vertebrate is right from where? Pisces, right? We came down to what? Amphibia, right? We came down to what? Reptilia. Then we came down to what? Aves. If you watch, remember the topic is evolution among. If you watch, this is how this is the trend. This is their evolutionary trend. From fish, come down, till back to down to what? Mammals. So the last one we look at is what? Mammals. Mammals. Look at what? Mammals. We look at what? Mammals. Or mammalia. Mammalia. So we go back to the first one. I said we look at. Another one is, yes, we go down to this first one, Kingdom Monera. Okay? We go down to what? Kingdom Monera. Excuse me. We go down to what? Kingdom Monera. Kingdom Monera. Kingdom Monera. As I said before now, in our previous class I mentioned prokaryote and what? Eukaryote. Remember when we treated cells, right? I talked about the eukaryotic cells and what? Prokaryotic cells. I remember. So this Monera, they are what? The prokaryote we're talking about. Okay? They are what? The prokaryote we are what? Talking about. They are the simplest living organisms. Sorry, our reference book for today's class is Modern Biology for Senior Secondary School by Sarojini Ramalingam, as revised by Lucy Akonwa and JBC Obidiwe. This is our reference book, revised edition 2018. This is our reference book for today. So most of the things you are going to see here has a lot to do with the content of this book. Okay? So we look at Kingdom Monera. Remember I pointed it out that under Monera we look at the prokaryote, bacteria, and what? Blue-green what? Algae. So we'll just look at their features, okay? Their features and the general features, and then we'll talk about bacteria and blue-green algae in a nutshell. Okay, now let's look at the Kingdom Monera as a whole. Are we together? Any issue, any challenge? You'll be going out coming, any challenge? Have you sent it? Send it now. Go and send it. Maybe you should miss it. Go and send it. No problem. Go and send it. Alright, so let's continue. So, the simplest of the living organisms, they are what? bacteria and what? Blue-green what? Algae. They belong to this kingdom. They belong to what? Kingdom what? Monera. Of course, they are very what? Small. Okay? They are very what? Small. And they are made up of what? I think I mentioned it, that their cells are what? Prokaryotic. They are made of what? Prokaryotic what? Cells. But there's not one thing there that they are enclosed in a rigid cell wall. I say that they are the smallest example of this kingdom. This kingdom, the organisms there, they are the smallest of living organisms. And they are made of mainly what? We have their what? Bacteria and what? Blue-green what? Algae. And I also say that their cells are prokaryotic. They are prokaryotic cells. And they are enclosed in what? They are enclosed in rigid what? Cell wall. Very important. Rigid cell. By the time you do your MCD, you do bacteriology. Yeah, I think so. MCD second semester. You talk about you come across bacteria. You see the cell wall. And if I want to study pharmacy, there are some drugs that target this cell wall. Am I competing? And when they destroy the cell wall, I'm taking you for let me come down. So, I'm just trying to point out the importance of this cell wall. To study the bacteria, they have cell wall. Cell wall. Rigid what? Cell wall. Of course, their cell is simple because they are small. It's a very simple cell. Now, there is no definite nucleus. There is no definite nucleus. No definite nucleus. So what does it imply? What it implies is that the chromosome is just free. Just for any particular part of the cell you can do with it. Unlike eukaryotic cells, where you have definite nucleus, okay? The chromosome is there in the nucleus, okay, right? But this one, there's no definite nucleus. So the chromosome is free and aligned in any particular direction in the what? In the what? The cell. So that's one feature of prokaryotic cells. They are small, simple, no definite nucleus. Now, of course, they are microscopic. Very important. They are what? Microscopic. Very, very what? Important. Know that they are microscopic. So this is the general feature of this. Not limited to this anymore. General feature of general features of this Monera. Now let's pick the two examples we have in our syllabus. Let's pick these two examples. Let's pick the bacteria and blue-green algae. So let's look at bacteria. Bacteria is plural, okay? Singular is what? Bacterium. Bacterium. Remember I said that the Monera, that they are what? Microscopic. Now, so bacteria can only be seen with what? Light, but not every light microscope. The one that are what? High power. Those the light microscope that are what? Have high resolution. Can see, you can use to see what? This bacteria. That's why if you go to microbiology lab, what you see there is what? Microscope. Two of us. Microbiology lab without microscope. That one microbiology lab. Microbiology lab without microscope. That one microbiology lab. It's not microbiology lab. So they talk about micro organisms. You cannot see with your naked eyes. Now, unlike virus, you know virus, can you see for virus with just light microscope? Can you see it? Light microscope, can you see it? What can you see it? Okay, electron what?
Electron microscope.
So let's continue. We are discussing bacteria. Let's continue. Now, they are found. They are everywhere. They are found in air, water, soil, even inside what? Living organisms. They are everywhere. Found in the No, if you are describing an organism, see the point you're going to, even though you're not writing essay, but these are the points you're going to study about any living organism. First of all, you need to look at the feature, the characteristic feature, the structure of the organism. Then also talk about the what is it called? Where the organism can be found. Okay? Good. Now, this bacteria can be found in the air, sand and what is it called? Even water, and even inside the organism. That that they're telling if you use sand to cover your injury, you have tetanus, right? So what is that thing? There is bacteria there, Clostridium tetani. Clostridium tetani is found in the sand. So if you use sand or soil to cover your injury, you can get infected. Okay? So it's Clostridium tetani. So that means tetanus came from. Okay? Tetani. Don't worry. You will see get it. Now let's continue. So that shows that bacteria is everywhere, even in the water you drink. If you're not careful, true of us. The air, there are some that you can get through air. You can inhale them, true of us. So they are everywhere. They are everywhere. So what is their size like? Their size is about 0.1 to 10 micrometer. Their size is about 0.1 to 10 watts micrometer. 0.1 to 10 microwatts meter. 0.1 to 10 micrometer. And they are in different shapes. They are in different shapes. We have the Coca bacilli or Coccus bacillus. Coccus bacillus singular. Coca bacilli. Coccus bacillus. Hope I got it spelling. Bacillus. Or Coca bacilli. Coca bacilli. Coccus bacillus. Coccus bacillus. This should double Coccus bacillus. Now, if they are spherical, you hear coccus. You hear us streptococcus, staphylococcus. Am I correct? So if they are spherical, that's where the name came from. Where coccus came from. Bacilli, if they are like a rod, you see a bacillus rod-like. The one that are like a rod. You know how rod look like now. So coccus bacillus. Basically, that's the two main. Coccus bacillus. So now there are ones that appear on their own. The one that come like chains. You see them like this, even though they are what? Cocoa. Something like this. Spherical, but they are what? Chains. The one that are on their own. This one is just coca. This one is the one that is in chains. This one is strepto. Strepto what? Coca or strepto what? Coccus. Strepto, coca. Strepto. This one is strepto. Coca. Basically, coca and coccus is circular. They are like this, in chain like this. Is strepto. You've heard bacteria that have streptococcus or streptococcus, right? Good. Then just sometimes you will see this coca like this. They are not in chain like that. They are not like this. Look up. They are like this. Staphylococcus is like this. Staphylo. You heard about it, right? Staphylococcus what? Aries, right? So, everything came from here, up below. Coccus, straight like chain, like this, as an agglomerate. Agglomerate is what? Is what? Staphylo what? Coccus. Staphylo what? Coccus. So the bacilli are rod-like. They are what? Rod-like. They are what? Bacilli. So basically that, and they, so if you hear that name, if you hear Streptococcus, you don't need to be told that, okay, it's spherical in shape, okay, it's spherical and what? Chain-like. Okay, this one is, okay, staphylococcus, okay, but it's still what? Coccus. Now, let's look at another thing. There's something else I want us to look at. I think I'll talk about being single and being in chains altogether. Okay. So I said that they can be what? They can occur singly. Know that point. They can occur what? Singly or in what? Groups or in chain. Look up. Single. Look up. Single. Coca. In group. And in what? Chain. Okay. They can occur single. I just use coccus to give an example. They can occur in singly, in chains, or in what? In groups. Okay. Now, bacteria. The bacteria move. Bacteria can be motile. You say that an object is motile. What does it mean? Motile. It moves. Okay. Motile. Does it move? Bacteria can be what? Motile. It can also be what? Non-motile. Know that point. It can be motile. At the same time, it can also be what? Some are non-motile. Now, those that are motile, if you see those that are motile, they have flagella. They have flagella. Like some bacilli, we have flagellated bacilli. We have flagellated. They're like this. Bacilli. You know what bacilli is rod-like, right? Right? Rod-like. See it. There's some bacilli. See how they look like. See them. See the flagella. Bacilli. Okay? There are some bacilli that are, they are called flagellated bacilli. Flagellate what? They are motile. Flagellated what? Bacilli. They are what? Motile. Flagellated bacilli. They are what? Motile. They are what? Motile. Of course, you know that the plural, the singular for flagella is what? Flagellum. Hope you know that. Remember? Okay. So this flagella enables them to do what? Move in the what? Water. Move in what? Water. Okay? Move in what? Water. So water is a medium through which they move. Yeah, that help them to move. Those in air can also move, but air will move and not be known. But this one in water, this flagella help them to do what? Move in what? Water. Now, we have discussed how the types of bacteria at this level, based on their structure, based on their appearance, under the microscope. We have discussed some of their features. We have also discussed their location, where you can find them, right? Then, let's talk about their nutrition. How do they feed? Do they feed? How do they get their nutrients? How do they get their what? Nutrients. Few of them make their own food. Few of them make their own food. Few of them make their own food. Of course, you know the process of photosynthesis, right? You know the process of photosynthesis.
Australia. You make use of photosynthesis.
Okay, how do you make your own food?
Thoughts they maybe they depend on higher organisms.
Not all of them. So few of them make their own food. Blue-green algae is there, even though yes, there are bacteria, but some of them make their own food. Okay. Which is happening now, what I give you. You have data, right? That's right. There's something I want to check. There are some that also undergo chemosynthesis anyway. You know what chemosynthesis is? Huh? What is chemosynthesis?
They mix
Huh? They use. Huh?
Composition.
You said what?
It involves decomposition in manufacture of food, not only photosynthesis. As in chemosynthesis, I understood that the process involved in manufacture of food by living organisms.
Yeah. Read this. Read it out fast. Loud.
Photosynthetic bacteria, PSB, including oxygenic and anosogenic phototrophic bacteria, are prokaryotes that are capable of carrying out photosynthesis. Oxygenic photosynthesis is limited to cyanobacteria, whereas anosogenic phototrophs are widely distributed among bacteria taxa.
Does it make sense to you now? Cyano what? So bacteria can actually, that was a few of them. You know, I don't believe in long story. Once any adjective, you see there is no long story. That's what I teach. Okay? Because science evolves, okay? I'm not monopoly of whatever. Whatever you don't understand. Once you check it up, you see it. Few of them, okay? Okay, check chemosynthetic bacteria. Check chemosynthetic. Check chemosynthesis. Chemosynthetic. Check if there's anything like that. Is anything like that? We have chemosynthetic. We have photo what? Okay, check what is chemosynthetic. Read chemosynthesis for us, loud.
Chemosynthesis occurs in bacteria and other organisms and involves the use of energy released by
inorganic compound. Good. That's the next point I want to make now. The first one, few of them undergo photosynthesis. Then the next group, there's some other group that use energy from inorganic compound. These ones are what? Chemosynthetic bacteria. Am I completing? Chemosynthetic bacteria. So they can synthesize, they can photosynthesize, and they can also what? Chemosynthesize. But most bacteria, most of them do what? Take in ready-made food, okay? Most of them take in ready-made food. Some are photosynthetic, some are chemosynthetic, some take in maybe ready-made what? Food. And how do they do that? Of course, you know that bacteria are what? Saprophytes, hope you know. They are what? Saprophytes. So sometimes in dead organisms, in dead matter, they will break down. Sometimes you see them, they will break down the food that is present in dead organisms and absorb it. These bacteria that do that are the what? Saprophytes. Are the what? Saprophytes. No, keep it on me because I this is how my plans are. Keep it on. You can always check it up. Because see, this type of studies make you to understand it better, appreciate it, and you can never forget it. Okay? You can never forget it. If they call it our next 2 years, 3 years, ask you about, you can talk about chemistry, but two of us, because you checked it, I allow you to check it yourself. So now, I say that they break down the food in the words, dead organisms, break it down, and they do what? Take the words, whatever they absorb, whatever is left in that organism. And these ones are the what? Saprophytes. They are the what? Saprophytes. Of course, you know how important saprophytes are in nature. Hope you know how important what they add to the soil. Hope you know. So we do that when we go into the nitrogen cycle, three of us, and the cycling of nutrients. Okay. So let us not go into that. So this, of course, this organism that died, the dead organism, once you see them decaying, decomposing, know that something is going on. True of us, mainly bacteria. Sometimes fungi can do that. Hope you know. But mainly what? Bacteria. Am I completing? Now, once you see the decay, and as they decay, they are adding nutrient to the what? The soil. So now, there are some that are face living organisms. The extra living organism and face the living organism and dealing with it, the living ones. They are called what? They are called what? Parasites.
What's up? I thought parasites are different from.
Parasite is anything that is inside you and feeding of you. It's a parasite. So far you are living, that is inside you, whether you are plant or animal, it's inside that organism and feeding on that organism. That is a what? Parasite. Please. Okay? Good. So a large number of bacteria, they live and feed on living organisms. They live inside living organisms and feed on them. They are known as what? Parasites. So we have parasites, saprophytes, chemosynthetic bacteria. Name them. The photosynthetic one, and what are they? Okay. So this is about their nutrition, right? Hope you understand how they are going now. Yeah, we just discussed their nutrition, how they get their nutrient. Because nutrition is a vital process, characteristics of any living organism. Very, very important. Because it's when you get nutrients, that's when respiration can take the two of us. If you have nutrients, so they are what break down two of us to release what energy. Now drive the process of the what? Living what? Organism. Now, so next, most bacteria reproduce by what? Binary what? Fission. We are now into what? Reproduction. That's why they multiply like fire. They multiply like what? Wildfire. They multiply, they are spread. Most of them reproduce by what? Binary what? Fission. Binary what?
Fusion.
Binary what? Fission. Binary fission. Binary fission. Most of them reproduce by what? Binary fission. And what is it all about? They divide into two daughter cells. And they are what? Identical to the parent cells. You don't divide into two daughter cells, which are identical to the what?
Parent cells.
So this binary fission, what type of reproduction is binary fission? What type of reproduction is binary fission? Eh? What type of reproduction is binary fission? Asexual now. Is it not? It's asexual reproduction. Okay, so let's continue. So I said that most bacteria reproduce by binary fission, divided into two daughter cells that are identical to the what? Parent cell. So this is a type of what? Asexual reproduction. Now the daughter cell will now grow and keep dividing. Okay? The daughter cell will also grow and also do what? Divide into two. Within few minutes, or minutes, or hours, or days, minutes. When you put that, the daughter cell that came out today, within few minutes, that one has divided. That's why they divide into thousands, millions. Few minutes, they keep dividing. So the daughter cell can grow and divide into two within a few minutes. So the rate of growth is very, very what? Fast. The rate of growth is very, very fast. That within a short period of time, you have millions of the bacteria in the living organism. And that's when you start seeing the effect. Their effect. When they are small, you do not see the effect, but they are dividing. When they become much, when the load is much, that's when the organism will feel it. Am I talking? Maybe if it's salmonella type, if it's salmonella type, when that causes typhoid, you see that with time, you know it's a bacteria, right? With time, when the person is down with typhoid, that means the load is much now. The body is now feeling it. But when you are staying, you do not feel, because human body is complex. Two of us. Human body is complex. The cells are billions, millions of billions of cells. So when they are divided, it must move until they become much that the body will feel it. That's when the person will be down. Now, if you go for typhoid, they'll check the tiger value. They call it tiger value. They want to check how is it in the system. The same thing, not only bacteria, it's the same thing with virus. They will check the viral load. Those that have HIV, they will check the load, the viral load, the amount of the virus in the body. That determines if the viral load is very high, like 10 days that person will be very, very down. Okay? And so let's continue. So, by the virtue of telling you that bacterial leak can be found in air, water, sand, it's clear that bacteria are found in many habitats. They can survive in many waters, as many as. So they can live in many habitats, even in the harsh ones. They don't care. They will survive there. Oh, I want to ask you a question. What is the name? What is the name of the cell division that occurs in asexual reproduction? What is the name of the cell division? What is the name of the cell division that occurs in asexual reproduction? What is the name? What are you saying?
Name of this.
The name of the cell division that occurs in asexual reproduction. Okay, let me ask another question again. What is the name of the cell division that occurs in sexual reproduction? Hey. Okay.
It's confusing.
It's not binary fission.
We know that binary fission is a form of asexual reproduction. I'm asking you a question. What is the name of cell division? It's water. How many cells? What? Somatic. What gives rise to the basic division you know that causes the big cell and the sex cell? What are they? Mitosis. Mitosis is the cell division. Of course, reproduction. You know, it's not said right now. It's the body. It's the joint is not divided into two of us. Into so it's my. Let's continue. It's okay. Let's go. If you are this, let's another area of importance. Very. You know, in biology, when you say economic importance, you talk about the what, both the positive effect, right, and negative what. Now let's take the importance of bacteria. Let's start from the harmful bacteria. They cause disease in what? Plants and what? Animals, including humans. I think I have mentioned some soil here, right? Yes. I have mentioned typhoid, right? I have mentioned tetanus. There's this bacteria also, Helicobacter pylori, that cause all into mainly gastric ulcer. There are many of them. When you come up into the medical, you understand better. But I just mentioned, I mentioned typhoid here, right? Even tuberculosis is caused by what? Mycobacterium tuberculosis. There are many of them. There are those that have pneumonia. There are many of them. So they cause a lot of what? Of course, you have felt streptococci, right? Those people that hey, come and see staphylococcus in this bottle. You can also see staphylococcus in the bottle. They are going to stay in the market. They will pack box. So you come and see staphylococcus that they just defecated. Come and see it in this bottle. You cannot see staphylococcus with your naked eyes. Come and see staphylococcus. Staphylococcus is not Igbo language. It's not true. These things, staphylococcus is a bacteria. Staph is a bacteria. Of course, staph is sexually transmitted. Hope you know. So this causes what? Harmful effect in the body. Of course, I mentioned the positive impact of bacteria here, right? Mm-hmm. When they cause decay, right? Mm-hmm. And they add nutrients to the soil, right? Mm-hmm. Is that a positive effect? So they improve soil what? Harmful effect, they cause diseases in plants, animals, even human. And positive effect, we'll talk about the what? They improve soil what? Because they are sacrificed. They cause what? Decay, right? They cause decay, thereby improving what? Soil fertility. They're also used in various industrial processes. Bacteria are also used in various industrial. In fact, there's this oil spillage. There's this oil spillage. Take care of oil spillage. There's some bacteria they release. They take care of this oil spillage. They mop up these things. I'm not going to. In fact, Google now, do it now. Use of bacteria in oil spillage. Let's see what you will get there. Check it up and read it loud. You know, people are watching us, so read out. So those that are our viewers can hear out, please.
So all the industry, like how did they get the bacteria? So now
you can culture bacteria now. There's what you call culture. You can culture bacteria and get millions of them you want within few minutes. Do you see anything like that?
Bacteria, products of bacteria. Most bacteria are good for bacteria in our bodies. They help degrade the food we eat. They help make nutrients available to us and neutralize toxins.
We know that one,
Let me see examples. As Also, they play an essential role in defense against infection by protecting.
Yeah, we have the one that's called normal flora in the system. We have bacteria we call normal flora. That's why there are some people, if you use too much antibiotic, it's not good for the system. Especially those that say that they are treating toilet infection. If you use too much antibiotic, it will go. You know, in the let me use female reproductive parts, vagina and what have you. The more the small thing you take antibiotic, if you use it too much, there's normal flora there that help to even protect the place. You use too much antibiotic, you go and eat flora. And when it's done, what's called supra-infection. Further infection will do what? Come on. And you see the person now having fungal infection because the normal flora has been shattered. Even inside our system, not just in the female reproductive system. Right. Did you see anything about use of bacteria in oil spillage?
They are important in sewage treatment and the breakdown of oil spills, the production of cheese and liquor through fermentation, the recovery of gold, palladium, copper, and other metals in the mining sector.
The one I'm interested in is that oil spillage. They need to mop up. So, bacteria is of importance there, okay? There are many of them. Now, let's continue. So, this is what we have to say for now. By the time we start answering past questions, we now see more on that. Basically, bacteria generally have a structure. If you look at this Nigeria here, you see the flagella is there. Remember I mentioned cell wall, right? Even though this is not a complete diagram. Remember I mentioned cell wall, right? The bacteria have cell wall. Okay. They have cell wall. They also have cytoplasm. They have what? Cytoplasm. But what they don't have is what? Nucleus, right? Good. So the chromosome is just free.
They don't have inputs. Yeah. I thought it's something like no definite.
You cannot say this is their nucleus.
Okay, that means that.
They are prokaryotes. You know, the chromosome is in structures. Do they have mice and chromosome guys? Stay anywhere you like inside the cell.
Okay, they don't have cytoplasm.
They have no, but they are okay. The best thing is they have no definite nucleus. So leave it like that. No definite what? Nucleus. Leave it like that, okay? So, next, remember we are talking about kingdom Monera, right? We've read so much on bacteria. Let's look at the blue-green what? Algae. Blue-green what? Algae. So where are they found? Where are the blue-green algae found? They are found, please, this is not the same thing with asparagus, I hope you know. I hope you know. So blue-green algae are found mainly in fresh water. They are found mainly in what? Fresh water. Then if you come on the land, they are found on damp places, damp places on the land. Fresh water and damp places on the land. Fresh water and damp places on the land. They are found in fresh water and damp places on the land. They have chlorophyll. So they can make their own food. Know that part. They have chlorophyll. And they can make their own what? Food. But there's something different from their own chlorophyll. Their own chlorophyll is not found in the what? Chloroplast. There is no chloroplast. Okay? Their own chlorophyll is not found in, unlike eukaryotic plants. Okay? Okay? You know plants are eukaryotic, right? But this one, their chlorophyll, yes, their chlorophyll, they have chlorophyll. They can synthesize their own food, but chlorophyll is not inside the water. It's there on its own. It's dispersed as particles in the center. So their chlorophyll is dispersed as what? Particles. What? Cytoplasm. So, just as here, they can exist singly on their own, they can also exist in colonies. They can exist singly on their own, they can also exist as what? Colonies. They exist singly as their own, or as what? Colonies. Examples, we have your words. Nostoc. We have nostoc, oscillatory, and amabena. Nostoc, N-O-S-T-O-C. N-O-S-T-O-C, nostoc. Oscillatoria from oscillatory, oscillatoria. Oscillatoria. From you know your oscillatory notion now. Oscillatory. We have nostoc. We have oscillatoria. We have what? You can look them up and check how they look like, okay? You can look them up. These are examples of what? Blue-green words. Blue-green words. You can look them up at your leisure time. I don't want to talk much on this. In bacteriology. Let's leave for those in the words bacteriology. Okay? So, but first, bacteriology is a different area of this course. A lot. That's why you not go deep into bacteria. So let's not go deep into that. That's why I try to limit myself by yourself. So we don't into that. Okay, now. So this is the enough we can take on the kingdom of Monera. Let's look at Protista. Kingdom Protista. So eukaryotes begin from kingdom what? Protista, okay? Kingdom Protista are what? Eukaryotes. Because in the secret question, the religion are eukaryotes. You see blue-green, you see bacteria, you see, they not even mention class, they don't want to kingdom. They pick examples of organisms in those kingdoms. This is which of these following is a eukaryote or a prokaryote? You see bacteria, you see amoeba, you see paramecium. So you know which is which, okay? So eukaryotes start from what? Kingdom what? Protista. Kingdom what? Protista. They are what? Eukaryotes. They are what? They have a definite what? Nucleus. And their cell structure is complex. To compare with what? Monera. Monera have simple cell structure. But the cell structure of this kingdom Protista is what? More complex. At the same time, they have what? Definite nucleus or true nucleus, as the case may be. Okay? Good. And they are larger than the Moneras. They are larger than the Moneras. they are larger than the what? The moneras. So these protists can be single cells. In fact, what can be? They are single what? Cells or unicellular organisms. They are single what? Cells. Have I mentioned that they are eukaryotes? They are single what? Cell. Have I mentioned that their structure is complex?
Yes.
And they have definite what? Nucleus, right? So note this point, okay? Of course, remember we treated cell organisms, right? So they have cell what? And these organisms carry out functions. They have cell organisms, and these organisms carry out what? Specific what? Functions. They have cell organisms, and these organisms carry out specific what? Functions. They have cell organisms, and these organisms carry out specific what? Functions. How many points have I mentioned now? This is a strong points, okay?
5.
5, right? Good. So, have I mentioned that they are bigger than the what? Larger than the what? Okay. They are even up to 10 to 100 times, like 10 to 100 times larger than the what? Moneras. So where do we see them? Where do we find these protists? Where do we find these protists? Most of them live in the water. Most of them live in the water. Like rivers, seas, and lakes in the deep. In moist places, even in the body fluids. Water. Example, rivers, seas, moist places in the body fluids. So these fluids are going to the water. Are you hot? Sorry about this. Here it's quiet, but it's hot. That's why it's not quiet, but it's not quickly quiet and noisy. Alright, so let's continue. So now, I say that they are found in rivers, seas, moist places, even in the body fluids of what? Living organisms. Please know this point, okay? Know this point. So another point to make is about their motility. They're motile. So they are motile. So most of them are motile, and they move by what? By the aid of what? Some have what? Flagella, right? Yes. Some have flagella, right? Some have cilia, right? Or cilium, right? Then some have false feet, pseudopodia. Two of us, with which they move. Okay. So some of them have flagella, some have cilia, some have what? Pseudopodia. These are three important structures that helps in their movements. Okay? Some have cilia, something like this, right? You can have cilia, you can have. Don't worry, we'll see what I'm just. Let's test knowledge. Because you can have. Is this cilia? What does this flagellum it has? Check well. You green out. Confusion. It's flagellum, no, it's flagellum.
Then Paramedics will make use of
cilia.
Since
How about use what?
pseudopodia.
Pseudopodia, true or false? Yes. Alright, let's continue. So we have to take, we talk about general things that they share in common. Now let's look at what is the similar for pseudopodia.
What is singular?
Pseudopodia.
Pseudopodia.
Cilia. Flagella.
Blue.
Blue. So now let us now look at this. Under this protista, I mentioned we have what? Protozoans and what? And what? Protophyta, right?
Yes.
And I said that protophyta have plant-like feature, right?
yes.
While protozoa have what? Animal-like what? Features. You can analyze in between for the two of us. That's animal-like features. So let's start with protophyta.
Okay.
Let's say one or two things. Remember here, we are not going deep. We just want to say one or two things so you can be able to answer questions there when you come up and university level. If you have interest in taking up a course in biology, as you wish, you can then go into these things. Alright, so now let's look at protophyta. I said that these are protists. I have plant-like what? Features, right? Good. So why do they have plant-like features? Why do you think they have plant-like? What do you think makes them have plant-like?
They have a synthetic.
What are the structures that make them to have plant-like features?
They have cell chloroplast.
Good. Cellulose what? Cellulose what?
Cell.
Wow, good. They have cellulose cell wall. Please note that point. They have cellulose cell wall. And they also have organelles called what? Chloroplast. So they have chloroplast and they have cellulose cell wall. Okay? And their chloroplast contains what? Chlorophyll. Remember, unlike blue-green algae, it has chlorophyll. That's why it can photosynthesize. But the chlorophyll is not found like what? Chlorophyll. But protophyta, their chlorophyll is inside the what? Chloroplast. Does it make sense? Good. Because you cannot have organisms, two of us, they have two cells. This is two what cell should be. Good. So chloroplast contains what? Chlorophyll. To do what? Inside their own what? Food. We have examples of them. We have their what? You are chlamydomonas. You have their what? Chlamydomonas. Their what? Chlamydomonas. Chlamydomonas. You have their chlamydomonas. You have their chlamydomonas. We also have chlorella. We have their chlamydomonas. We have their chlorella. Chlamydomonas, chlorella. Yeah. Chlorella. These ones, these chlamydomonas and chlorella. Remember I said that these are found, you can find in fresh water. What they're talking about their general feature, remember? Fresh water. These chlamydomonas and chlorella are found in what? Fresh water. Please put that down. Fresh water. Water. Another one is diatoms. Another one is diatoms. So, protista, we have, for example, chlamydomonas, chlorella, diatoms. Chlorella and chlamydomonas are found in fresh water. Diatoms are found in both in fresh water and sea water. Diatoms are found in both in fresh water and sea water. Diatoms are found in both in fresh water and sea water. There is one peculiar feature of these diatoms. They have silica coats. Silica coats. So they are coated with what? Silica. You know where silica came from now? True of us. So they have coat made up of what? Silica. So the diatoms, they have silica what? Very hard what? Silica coats. So in a nutshell, this protista, they produce their own food. They have chlorophyll found inside the water. Chloroplast. Okay? Chlorophyll inside the water. As against the blue-green algae. These things are salient point too. Now let's move straight forward to protozoa. Let's move forward towards protozoa. Protozoa, they are protists that have animal-like features. Protozoa are protists that have animal-like what? Features. They have features that makes them look like what? Animal. One is that they feed on already made food. They do what? Feed on already made food. And they have no cellulose cell wall. They feed on already made food and have no cellulose cell wall. They feed on already made food and have no cellulose cell wall. Okay? Good. Examples here, you know them now. Amoeba, right? Paramecium, right? So we are no longer alien to them. They are examples. We have the amoeba and what? Paramecium. In fact, we have even seen this amoeba finish. We have described them even in this class. Two of us, we described their locomotion, a lot of things about them. So we wouldn't talk much on that. Except unless when we go into cell, different levels of this thing, we cannot talk about them. Now, have I mentioned that we have the one that are put that have animal-like and plant features, right? When I mentioned the winner, right? So, do you know why? What makes them to have animal-like and plant-like features? The winner has what? Chloroplast. Know this point. This is a jam question. They ask this question in jam. The winner have chloroplast. But it does not have what? Cellulose cell wall. Euglena has chloroplast. Yes. It's shocking.
What now made you plant this thing? I thought they said it has several. What made you bring out that plants?
You know the plants, plant-like feature. It has chloroplast. That makes it plant-like. Remember, it's not plant, it's plant-like feature. You can't call it plant. It has plant. It's Monera. Plant is a different kingdom. Protista is a different kingdom. Two of us. You know, initially we have two kingdoms. Or just animal kingdom before they say no. It's 5 kingdoms. So they arrange all this. Protista is a different kingdom on its own. That's the different one. But it has plant-like feature, not plant. Plant-like feature. So the plant-like feature you have is what? Chloroplast. Good. But animal-like feature is that it does not have what? Cellulose. Does it make sense? It has no what? Cellulose. Does it have eyes?
Yes.
Does plant have eyes? Eye?
No.
Does plant? It's also an animal-like feature. Two of us. If you go into the cell, right? Now, so what we have is plasma membrane as against what? Cell has the plasma membrane called pellicle. You see jump is pellicle is the plasma membrane found in what? Pellicle. Please note these things. Pellicle. In what? The greener. So it's an animal-like what? Feature. However, there are some greener that can make their what? That take in already made what? Food. There are greener that take in already made what? Yes. So what does it mean? Those ones are colors. What are they?
Yeah, they're not specific.
Good. They make their own food. There are some million and million, but they are few that make their own food. Colorless. You can take it up. Do it fast. Let's see what we have. We are describing this what is it called? The adhera feature of this phylum, the protozoa. So let's look about it. I'm missing anything.
That's making that both endotropic, most conditions are endotropic.
You say what?
Make its own food.
Let me see what is it.
Mesophyte.
Different types of green, okay? We just type colorless urina. Let's see what we have. Colorless urina. Colorless urina. Nothing much is here anyway. Nothing much is here. Nothing much here. Let's continue. If I see any information on that, I'll let you know. But there are ingredients that do not make us that feed on already made what? Food. Let's not waste time. Continue. Now, produce. How do they reproduce? You're not sure what it's not the same. And just tell me how they produce.
They make use of like paramezone, make use of
So they reproduce.
yes, which is
What I say?
yes, they produce sexually and asexually.
Confusion is part of the answer. I assume myself is part of the you see that
Produce sexually and asexually.
sexual reproduction.
No, that's not it. So
Ah, Oh yeah.
And then
What's this say?
that paramecium produce sexually and asexually.
Do you agree with that?
Is it paramedic?
Paramecium. Is that okay?
Yes, beginning
Etc.
yes. But sometimes they do that amoeba do
Sexual. Do what?
I'm not going to say
Ow! Ow!
English.
Do process. Sexual.
Which one?
Ameba.
I'm not going
that is asexual.
I'm not going to say etc.
Paramecium is what? Alright, so they mainly, so some of them are reproduced by what? Asexually by what? Binary fission, okay? However, many of them reproduce what? Sexually. To produce male, of course, you know what happens in serial production now. Male gamete, right? Male and female gamete come together to form zygote, right? Then after formation of the zygote, other things come up, okay? Meiosis, blah blah blah, to form spores, right? So, and each of the spores do what? Develop into what? New what? Organisms. Good. Okay. So this is, remember we are talking about evolution. Remember, evolution, the topic is what? We are not just, listen carefully, we are not just treating this organism. No, we are talking about evolution among these people. And this is an evolutionary trend from Monera to Protista. From not having a true definite nucleus to having a definite nucleus. True of us. From not having chlorophyll inside chloroplast to having chlorophyll. I'm not commenting that. So you see, it's a trend. Now, under this Protista, sexual evolution has only what? So this sexual evolution that entered now is an evolutionary what? Advancement. Okay? Okay? Good. That's the point I want you to make here. And as you move from this Monera now to fungi, you start seeing as you move from Protista to fungi, you start seeing more evolutionary what? Advancement. Okay? Good. So these are the things I want you to know, to point out, to always to observe in this our class. So I want to finish with Protista today. So next lecture we'll start with kingdom fungi, okay? It's almost 2 o'clock. And we're losing light. So let's go. So let's now look at the classes. Let's look at the classes and do what? Wrap it up. Okay? Yeah. Classes under. You know, we just described the general, okay? I just what to place these organisms I have been calling under different words, classes. So we have the one we call Rhizopoda. 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 We have the one we call Rhizopoda. Rhizopoda. This Rhizopoda, they move by what? Means of what? Pseudopodia. Example is ameba. Rhizopoda move by means of what? Pseudopodia. The example is what? Ameba. The next one we have the Celiopoda. Celiophora, sorry, not poda. Remember our reference today is this, so most of the things we are saying is from here, with additional things I say. They move mainly by cilia. Example is what? Paramecium. Paramecium is under here. Okay? Okay? Paramecium is under here, move by what? Cilia. This one, ameba is under here. Another one is Mastigophora. 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 This one, flagella, is one at work here. Flagella is one at work here. Flagella is the one at work here. Flagella. An example of this is Trichomonas. Trichomonas. This one they call the disease. This Trichomonas. Trichomonas. Cause disease. Trichomonas. Check the disease cause of Trichomonas. Let's see what we have here.
I need to also
I like green.
Even like this will be like confusing, okay? Okay. But don't worry, just check what is there first. Let's see.
This is caused by
Trichomonas. Do you have the data? Let me see. Caused by Trichomonas.
in females. Trichomoniasis is a common cause of vaginal discharge and is associated with poor birth outcomes and increased risk of pelvic inflammatory disease in girls.
Caused by what? Trichomonas. Open it. Colon first. Go to colon first. So, did you see any other disease caused by Trichomonas? What is this vista all about? Okay.
Types of
What do you do?
That was what I was studying.
What do you do? What task do you do?
Just if you don't know something and then if you perform other tasks, prepare the tasks, take on them every day.
What type of task is that?
Various tasks and other posts.
Okay. Okay, yeah. Why are you doing those like that? Huh?
interested.
Am I interested?
Last 10 days of today is the 10th day. They started sharp. So they
Okay. Ubina is on ubinoidea. Ubinoidea. That's where it belongs to. You know, remember, see, what you are doing here is just simple at this level. Okay? We are not doing deep, okay? Just let's work with this for now. It's not onomastic, it's ubinoidea. So
That would know that it's under what?
yes, it's a class. Protista is phylum. Under phylum we have class, remember?
So it would
It will know that.
that's of protista.
So let's continue, please. Let's know what's time. So we have sporozoa. 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 Under sporozoa we have their what? You have their what? You are plasmodium is here. Plasmodium is here. Plasmodium is under the what? Here. Plasmodium is here.
They move the end of what?
What does plasmodium cause? What is this? Thank you, malaria. Thank you. It causes what? Malaria.
Whereas there's a good that it moves with.
Huh?
It moves to the end of what?
What's that?
Sclerosis.
No.
Nothing.
They have no reason for movement. You know, it's a vector that transfers it now. What is that vector? What vector transfers Plasmodium? Mosquito. That causes malaria, right? It's mosquito now. If I'm infected with Plasmodium, if you suck my blood, it will enter the mosquito, it will go to another person. True or false? In trying to feed on the person's blood, it will enter the system. True or false? So,
is that the only way malaria is caused?
Someone that
Or is it just the form of transfer malaria?
what other form now can you
Because I thought that malaria is washing away of the old red blood cell.
No. Malaria, no.
Sir.
Now you said I will not call it malaria, but this kind of thing, suppose malaria is
No, it's not.
What about if Maria and mosquito did not bite and you are still suffering from malaria? Sometimes they say that those that have the genotype.
An African man, sorry to say, basically,
Yeah.
as you are now, if I take your blood, sir, and go to lab, I must see some trace of Plasmodium. You know, malaria, Plasmodium, mosquito is here. But see the point. I may not have clinically infected, but you may not have malaria. You start having clinical malaria. You know you have an immune system. True or false? Good. If they are work, true or false? Good. And stress, you may have Plasmodium in the system, but you may not have signs of malaria. But sometimes stress may bring down your immune system. It will surface. I'm not going to. That's also why they say, someone, what do not bite? Possibly there's Plasmodium in the bloodstream, but because in the system, but because of the immune system, or even if you stop eating well, stop taking enough fruits, the one in the system may come up. Am I correct? So, and you start having clinical malaria if they destroy, of course, when the red blood cells are being destroyed to the extent that the debris that is in the system will become much. Fever will start coming in, and other clinical malaria, other symptoms of malaria will start coming. This is a very important, interesting area I don't want to go into. If you watch, I'll be trying to do it chap. It will distract us. Maybe I think we'll still treat malaria in biology. We will with that topic. I think there's a topic like that in biology.
I was told that it is also the old red blood cell.
It's not true. Not true. So I think we have done justice to this. I mentioned spores, right?
Yes.
Formation of spores as a form of what? After cell reproduction, right?
Yes.
Zygote, right? Spore
Yes.
releases the organism, right? Another thing. So this is also an advancement as against what? Monera. Okay? I think we'll stop it for today. Shall we?
Yes.
Alright, thank you very much. I don't know if there's another question, we'll stop here. We'll continue tomorrow. So I want to welcome our viewers. Thank you very much for being with us in these tutorials. Tomorrow again, we'll continue tomorrow with chemistry. At exactly 4 p.m. 4 p.m. by the special grace of God. We'll be here again live. We move over to chemistry. That will be tomorrow. Be with us again tomorrow. And God will bless you as you do so. Please follow this page. Subscribe to our live subscription so we can take this forward. Share with your friends. And God will bless you as you do so. See you tomorrow. All right. God bless you.